currently developing a strategy to search primarily for ancient life during a wetter phase of Mars. Perhaps we need to also think in terms of present-day life still holding on somewhere in the subsurface. And again, I chose the word prudent carefully. I do think it's time to start to think about active processes as well as extinct processes.
Just to add to that, one of the things that's been in our strategy for what we should be doing at Mars is what we've termed a Mars Science Orbiter. And part of that was, in fact, to measure trace gases in the atmosphere of Mars there around Mars, so that you could do a sufficient resolution so that you could find where methane may be seeping out of the subsurface. So what that does, it gives you the opportunity to actually determine where to go so that then you can make more germane measurements right there at the spot where the gases are coming out.
Yeah, I'd like to comment on that, if I may. I've thought a lot about, independent of budget issues and political issues and current planning, what is the right strategy for exploiting this discovery? And it seems to me that demonstrating for the first time that we have regions of active release is key, and demonstrating that lifetimes are short and so on means that we really need to monitor. I wouldn't say monitor, that's a bad word in science, but we need to measure Mars' surface all the time. We need a way to identify where all of the active vents are, to quantify their principal gases, to identify which might be dominated by biology, if so, which might be dominated by geochemistry, and then based on this identify those few, establish that they are repeatable from year to year or not. And then this leads to a data cube or data polygon or whatever it is that permits you to decide, okay, this particular site is where we really want to go, put down a higher resolution mapper and then maybe a probe and so forth and so on. So I think what we're really seeing here is a window into a strategy for that class of evaluating the significance of these releases that we don't currently have in our suite of instruments or capabilities. We need something new.
Okay, we're going to take another question from Associated Press. Seth?
Yes, Seth Bornstein again at AP. Thanks for doing this. I guess for Dr. Pratt or Atreya, as you said that seems to be, the release points seem to be somewhat associated with areas you would think have more likely to have water underground. If you look at that combination, and maybe I didn't hear this right, is it more likely to be biotic or abiotic, biological or geochemistry? And can you explain why it would be more one than the other, the combination?
I don't think we have a compelling bit of evidence right now to say which one is more likely. I think, as we've pointed out a couple of times here, the takeaway message is that it appears to be an active process. It appears to be something which releases a seasonal signal, but that could be the long-term, it could be a process that's always ongoing and is only seasonally released to the atmosphere. We need some other lines of evidence before it really tips the scales to one side or the other for water-rock reaction, abiotic, versus an actual reaction that we would consider to be a form of life. I wish I could tell you that I thought it tipped the evidence one way or the other, because obviously as a life scientist, I would love to think that it's biology, but I just don't think we have the right line of evidence yet.
Geronimo might want to say something about this, since he's looked pretty hard at these areas. And
yeah, one of the things that when we see these localized areas, the regions we see methane appear to have hydrated minerals. And what are hydrated minerals? Minerals have been affected by liquid water. So these are important regions on Mars. And one of the ideas also could be that also some of the regions we see methane, we also see volatile rich ices probably below the surface. So the methane that we see could be being produced now or anciently, but it's being released now. So methane could be stored in ices. So we had to learn more about the isotopic signatures and see when it's being released and from where, more precisely, to better understand if it's being released right now, if it's being produced right now, or being produced before.
One thing that we, as a group, have talked about a bit is that this process of serpentinization, this simple water-rock reaction, it's a process we see only in a few very special places on Earth, usually associated with major fracturing and faulting that allows mantle-like materials to be exposed to seawater or groundwater. Also, that reaction is one in which there's a volume increase in the solids. So that's a process that tends to plug up the plumbing. It tends to isolate the reactive site. And again, if we look at Mars and we look at the landscape on Mars, we don't see a lot of evidence for major, active, deep faulting and uplift that would bring these reactive materials into contact with water. So if there was sort of a line of evidence that makes me think we need to seriously consider biology, it's that the processes in the subsurface that would allow for methane generation seem slightly more plausible for biology than geochemistry.
Okay, we'll take another question from Kelly of Sky and Telescope, and then we'll head down south and take a question from the Kennedy Space Center. Kelly?
Thanks for the chance to ask a second question. Mike Buma, in your paper you say that you actually searched for both methane and water and saw both. If we're talking about methanogenic bacteria here, they need the radiolysis of water to create the hydrogen in the first place. Would you see in your water signature any kind of deuteration of that hydrogen or something that would lead you to suspect that there's something going on there?
I'd like to ask Dr. Villanueva to respond to that.
Yeah, we have the beauty of our maps that we measure water and methane simultaneously. So we have the two signatures there. In this particular paper, we have focused on methane, but we are working on the data, and soon we're going to also present the two results, how they compare. On the basic raw analysis we see that there are regions that the two gases appear in enhanced. So regions where the methane and water appear localized, and some regions where water only appears by itself. So there are definitely things we need to learn more about. And one of the questions you just asked is about the isotopic signatures of water. And this is a very important chronometer on Mars. I mean, it's used to track the history of the water. So one of the things we are doing now, going back to those data sets, we have the data, is to see how old the water that we're seeing, maybe we can associate it with the methane and have an idea about how ancient this gas is. And we are thinking of submitting this paper very soon. So we're going to talk about this later.
Okay.
May I say a few
Sure.
Yeah, I just wanted to comment that following up on Lisa's comments and others, that if one does have a biome near the surface of Mars, then it should be using water that is in contact with the atmospheric escape, which of course is one of the objectives of the upcoming MAVEN mission. And we know already that the deuterium content in that water is very high compared with Earth due to the escape of the lighter hydrogen fraction. But as Geronimo was commenting, if we have life from below the permafrost, that water is ancient. It's been there a very long time, and it might be much lower in deuterium, as, for example, we see in Mars meteorites. And so this gives the idea of a diagnostic signature for the depth from which the methane is being released if you measure co-released water and, as you say, measure the T2H ratio. Yeah.
As well, we know almost nothing about the hydrogen-deuterium ratio on hydrogen that's generated by radiolytic splitting of water molecules. That might be a rather distinct signature. Very few experiments have attempted to measure that, and it's one where we need to do more work in our labs on Earth in preparation for recognizing the signals that we might detect on Mars. Because on Earth we tend to have processes like that masked by the overwhelming abundance of other processes that compete with it. But on Mars it might be the best game in town and might be where the action is.
Okay, now let's go down to the Kennedy Space Center for a question and follow-up, and then we'll come back to headquarters. Kennedy?
Thanks very much. This is Todd Halverson of Florida Today.